we go. Wonderful. Okay, thank you so much for joining me today. So, hello everyone. Welcome to the first episode of my series of science podcasts. And today I am honoured to welcome Professor Pamela Bjorkman, who is the David Baltimore Professor of Biology and Biological Engineering at Caltech. And she is the Executive Officer for Biology and Biological Engineering. So thank you so much for joining me today.
Thank you.
So, first of all, if you could just give us a brief outline of what your work involves.
Okay, so my lab is interested in understanding how viruses and other pathogens are eliminated by the immune system. So the way we approach that is to look at, for example, how antibodies recognize viruses by determining three-dimensional structures of the parts of the virus that the antibodies might recognize.
Wonderful. And so we're going to jump straight in to the technical parts, and on the podcast I will put links to the papers that I'm referencing to, and so the viewers can read if they want to and if they want to find out more. And so first of all, please may you explain what proteins related to Class I Major Histocompatibility Complex are.
Okay, so, well, the so-called classical Class I Major Histocompatibility Complex proteins, which I'm going to abbreviate here as MHC proteins. So Class I MHC proteins are on the surface of almost all the cells in our bodies, the exception being red blood cells, which don't have a nucleus. But what they do is that they present pieces of pathogens that may have infected a cell. So if something, a virus or a bacterium, if it gets inside a cell, it gets broken up into small pieces of itself through normal cellular processes. These are called peptides. And the Class I MHC molecule presents those peptides to the outside world. That is, it goes to the cell surface with those peptides, and then the immune system looks at the peptides in Class I MHC molecules and decides if it's dangerous or not. If it's dangerous, it kills that cell. So this is very useful because in the case of viruses, it prevents virally infected cells from turning into virus factories and producing more viruses.
Absolutely, thank you very much. So once you published your postdoc work in 1987, you ultimately solved the first crystal structure of the MHCs, which was the HLA-A2 human histocompatibility antigen. What further progress has come of it since your postdoc?
Well, there are a lot of structures since that structure. And, perhaps interestingly or perhaps not interestingly, they all look the same. So one thing to explain is that people are, the major histocompatibility complex itself, that is the genome, the part in your DNA is the most polymorphic genetic locus in mammals. And so people have different alleles of MHC1 molecules, and there's another one called Class II, but I won't get into that. So you have probably 6 different alleles of MHC Class I. There are 3 loci that encode them, and you probably, because they're so polymorphic, got a different one from your mother than from your father. So you have between 3 and 6, likely you have 6. And so these molecules have been linked to disease, so that if you do a typing of someone, you can sort of list like what are the diseases you might be likely to get. And some of these are autoimmune diseases, so that there's kind of a molecular explanation for why MHC molecules are linked to autoimmune diseases, which is that when they are recognized by T cells, which are the lymphocytes that actually recognize MHC molecules, they the T cell receptor recognizes part of the MHC molecule and the part of the virus, which would be the peptide, at the same time. So you can sort of intuitively understand that sometimes the immune system makes a mistake because it's always presenting peptides of its own proteins. So the mistake is, here's your MHC molecule. It's got a so-called self-peptide most of the time. And then every once in a while, there'll be a viral peptide. And the T cell receptor just happens to recognize the self-peptide with the MHC as something dangerous, and it goes and attacks your cell. So I would say there's been a lot of progress in understanding those links through these structural studies and just understanding more about T cell recognition and how this might be used in certain types of therapies.
Amazing. Yeah, that's absolutely wonderful. So I've researched a little bit into you, and it said that you've used 3D imaging techniques such as electron tomography and fluorescent microscopy to investigate HIV or SIV in primates, infection and in human tissues, HIV in human tissues. So how have you used these to your advantage?
Okay, so we're interested in HIV and how the immune system attacks it, which it does a pretty bad job because people who have HIV infections are severely immunocompromised. And the problem with HIV infections is that HIV infects a type of a T cell that is normally going to regulate your whole T cell response and your B cell response, which is what makes antibodies. And so you wipe out so-called CD4 T cells when you have an HIV infection, and then you're just, the person's immune system is very compromised and they can't make immune responses against normal pathogens that are just around us all the time that we would just fight off and never even know it. So what we were interested in is what's the HIV distribution in the tissues. And so to approach that, we used electron tomography, which is a form of electron microscopy, but it's in 3D instead of 2D. So the analogy would be if you go in to get an X-ray of your bones to see if you've broken something, they'll do an X-ray from one side, and it's a two-dimensional projection of all the bones. And sometimes you can't really see if the break is somehow not from a certain angle, you might not be able to see it. But then if you do a CAT scan, which is computer-assisted tomography, they take X-rays from all different angles. And then you build up a three-dimensional reconstruction of the bones, and you can see the break. You can see a lot of other things too. So in the same way, if you do a normal, you take an electron microscope and you look through it and you get some images. Those are 2D projections. But now if you tilt the stage that your sample's on, you now through some computation can build up a 3D image. So what we've done is look at where are the HIV variants in an infected tissue? Like, are they spreading around a lot? Answers like that. And then we combine that with fluorescence microscopy, which is lower resolution, so you can't really see individual viruses, but if you label parts of the virus with a fluorescent molecule, you can see very large areas in the tissues. So we try to combine a large amount of tissue at lower resolution with a smaller amount of tissue at higher resolution, and both of them are done in 3D. So we're just looking at the distribution of the viruses.
That's fascinating. And I suppose because of there's so many strains, and that's why it's so difficult to make a vaccine. Yes. Probably impossible.
Absolutely. If HIV
is huge in the advancement in therapies towards it, isn't it?
That's exactly why. I mean, people are saying, well, how did people manage to get a vaccine against SARS-CoV-2 so quickly? Well, fortunately, it doesn't vary a lot, even though people keep hearing about variants. But its rate of mutation is nothing compared to HIV.
Yeah, definitely. So my next question is what hope do you have for the future of broadly neutralizing HIV-1 antibodies in therapeutics, and how could we go about improving them in the future?
Well, if there was enough money, people could get infusions of these antibodies to prevent infection. You'd probably have to use, well, you couldn't use just one because you'd have mutations that would affect things, but you maybe if you use 2 or 3 and everyone in an area of high infection rates had these infusions, and you can do things so that the antibodies will last longer than they normally would. So maybe you could get twice yearly monoclonal antibody infusions. I don't think that's really going to happen that soon because just the expense and the logistics of it. There are things that could solve the HIV problem absolutely right now, and that's delivery of small molecule drugs in something called PrEP. So you just give someone, I mean, this is in use in the United States, and I imagine in Europe as well. People are given a dose of an antiretroviral drug, and that prevents them from acquiring an infection in the same ways the antibodies could, but they'd have fewer side effects. But PrEP is not worldwide, and there's just societal reasons for that, and perhaps financial reasons, and it's a immense tragedy because we shouldn't have millions of people infected with HIV today because there's a way to stop it.
Absolutely, and with the COVID-19 pandemic that we're in now, hopefully with the vaccine rollout, and people have recognized that it's a global problem, and so everyone needs to work together, but countries are protecting themselves first, which
that's
the right thing to do. So hopefully we can learn from that, and after this is most people are vaccinated, we can move on to tackling HIV and putting more effort into.
Well, I think they're connected, and here's the reason. In countries like South Africa, there are high rates of HIV infection, and those people, if they're not treated, are immunocompromised. And so if they also get a SARS-CoV-2 infection, that virus can remain in an immunocompromised person for a lot longer than in a person with a normal immune system. So that gives it time to make variants. And some people have speculated that the reason we're seeing variants around the world right now is that a few immunocompromised people incubated the virus for months and months and then infected other people, and now these variants are taking off. So if the world doesn't solve the HIV problem, there may be an issue with solving the COVID problem as well.
That's fascinating. It really puts things into perspective in what we need to do to tackle these and how so many things go hand in hand. And so my next question is, you've also done research into the vesicles and into how our neonatal FC receptor immunoglobulin antibody complexes delivered in intracellular vesicles to their destinations within a cell. So can you just elaborate a little bit on this?
Sure. So that was another electron tomography project where we were doing this 3-dimensional EM. And what we were interested in is a molecule called the neonatal FC receptor, which happens to fold into a 3-dimensional structure that resembles an MHC molecule. That was my original interest in this. But instead of binding a peptide, it binds to an antibody of the class called IgG. And one of the things it does is it transports IgG from ingested milk into the bloodstream. So actually in humans, most of the antibodies in breast milk are IgA. But in humans, this same molecule, the neonatal FC receptor, transports maternal IgG across the placenta to the fetus. So this is a molecule that looks like an MHC protein, but its job is to give newborns or fetuses maternal antibodies, and this protects them prior to the development of their own fully functional immune system. So we were interested in, if you have an epithelial cell barrier, the way you would have separating ingested milk in the gut, with the other side would be like the bloodstream. How does it go across? And so that's what we did, three-dimensional EM, to look for what types of little tiny vesicles in the cell have this. And so we were interested in mapping that out.
Yeah. So did you find that there were any specialized vesicles or a defined temporal path for this transcytosis?
That's an interesting question, because it was entirely chaotic. I went into this thinking there would be a defined path, and we left it thinking that it was totally chaotic. And in fact, I had a slide that I made when we were studying this. This was like, I think we published the final paper on the, well, we published since then, but the most complete paper was in 2008. So what we did was make a slide for a review article later that showed, how do you traffic if you want to move a passenger from one side to the other? So one side would be ingested milk in the gut and the other would be the bloodstream. Do you do it by going on defined pathways in a certain type of car? And so we had all these cars inside the cell, and we had like the IgG antibodies were represented by, people in the cars. And so that would be one way of doing it. So it would be like, here's the highway that you would go on, and you take this branch out and everything. And then we had another possibility, which was like, the cars are all over the place. Some of them are trucks with cars on top of them. They go all over the place, and somehow they wander around until they get to the other side of the cell. And it was the second one.
So there needs to be more research into that area, possibly.
Yes, that would be interesting. Yeah.
Okay, fascinating, because that might transfer into other areas as well, and it might.
Well, it does because another thing the neonatal Fc receptor does, its name is neonatal, but it's expressed throughout life. So that's an old name. But another thing that it does is to prolong the half-life of IgG antibodies in the blood. And this is incredibly important because therapeutics, like for cancer, antibody therapeutics for rheumatoid arthritis and so on, they're injected and they last a certain amount of time. But if you get them to interact in a better way with the neonatal Fc receptor, they'll last longer. So right now there are companies have made mutations that increase the half-life of IgG antibodies, which is the reason that some of these actually don't have to be injected. Instead of being injected every month, they might be able to be injected every 6 months, and that's a huge difference.
Yeah, amazing. So my next question is, you've then done studies on prostate cancer. So how may your structural studies of the prostate-specific membrane antigen lead to development of chemotherapeutics, cancer imaging agents, and agents for treatment of neurological disorders as well?
So you've really done your homework. I have to say that we don't work in this area now, and we did publish a structure of prostate-specific membrane antigen, and the reason was related to our interest in MHC-like protein. And so, to cut a long story short, there's an MHC-like protein called HFE, which regulates iron metabolism. Okay, and to do that, well, it's still not clear how it does that, but what it does is it doesn't bind peptides or present them the way MHC molecules do, which is a specific function in the immune system. Instead, it binds to transferrin receptor, which is what binds iron-loaded transferrin. Okay, so we were interested in how HFE interacts with transferrin receptor and so on, because It's an MHC-like molecule, but it's not binding to a peptide. It's not binding to a T cell receptor. So how does it bind to this iron, a receptor for iron-loaded protein? Okay, so now PSMA, prostate-specific membrane antigen, looks like transferrin receptor.
Okay.
So that's why we were interested in it. Like, how is it? That one's actually an enzyme, prostate-specific membrane antigen. It's a peptidase. It cleaves proteins, but transferrin receptor is not. So seriously, we were just really interested in the structural point of view of how is it that you have two proteins that adopt the same fold but do something completely differently. So our interest in that was to figure out, like, why is transferrin receptor not an enzyme but PSMA is? And so we're not working on any form of cancer therapeutics in that respect, but I think my interest is more this general one, which is, what does it mean when proteins adopt the same three-dimensional fold but function totally differently?
Yeah, absolutely. I mean, that's fascinating because that can be applied to so many different things, because I'm sure that's not the only one where
not.
The tertiary structure is
absolutely not. And it sort of is a warning for people looking at structures. The same fold can adopt many different functions. And so when I talk about this, I show this slide where the MHC structure, in the paper we published in 1987, there's a figure showing what it looks like. And in many people's minds, that looks like a moose. It's got like these antlers and stuff, and people have made all these funny analogies relating them. And so I show this slide that shows the MHC molecule in a field of grass and then the moose in the field of grass, and they look very similar, but obviously they have different functions. Now that's just a joke because the moose is clearly bigger than the MHC molecule, but some molecules adopt the same fold and then do different things.
Yeah, that's wonderful. And so my next question is, I'm not sure how to pronounce this, you'll have to correct me, but the Drosophila mutant Methuselah, or MTH. We're going to call it MTH for now.
Methuselah we worked on, and again, this is something, every once in a while, someone will come to my lab with their own interests. And this was the interest of Anthony West in my lab. And so we did a structure, and we're not following through on that. But let me just explain that at Caltech, Seymour Benzer was a very famous Drosophila geneticist, and he had isolated a mutant that conferred longer life to Drosophila, the flies. These are flies that are used commonly for genetics, and so Methuselah, of course, was the person in the Bible who lived for so long. So Seymour named it Methuselah, and so we did that structure. But again, we're not Drosophila geneticists.
Okay, that was fascinating in that it increased the lifespan by 35%, and so I thought that was really interesting. But
well, maybe people have followed up on that. There's a lot of lifespan studies being done in the model organism of worms. So but this is not my field, so I shouldn't really comment on that.
Yeah, well, thank you. And then my next question is, how do pore-forming toxins, which are, to the listeners, potent cytolytic agents secreted by pathogenic bacteria that protect microbes against the cell-mediated immune system by targeting phagocytic cells. So how do these disrupt the epithelial barriers and liberate the materials necessary to sustain growth and colonization?
Okay, so again, here I'm just, we don't work on anything directly related to this, but my understanding would be from structural studies of such toxins that they insert into the epithelial cell membrane to form some kind of pore that allow things to leak out.
Okay. As my next question is, please may you tell about your recent coronavirus work and your nanoparticles linking the 8 different strains and the possibility of a vaccination.
Okay, so when the pandemic shutdown happened in the US, which would be March of 2020, we had actually been working on looking at the structures of antibodies from COVID-19 patients a month earlier. So in February 2020, our collaborator at Rockefeller University, Michelle Nussenswag, they had gotten donors, COVID-19 donors from the New York area, and they were isolating monoclonal antibodies. Now, we'd been working with Michelle's lab for about 10 years on isolating HIV derived or antibodies from HIV infected people and also Zika infected people and so on. And so we had a long-standing collaboration with them. So in February, they switched to doing the COVID-19 research, and we got the antibodies in March, I believe, or April. And then Christopher Barnes in my lab started this incredible effort to just solve as many structures as we could of those antibodies bound to their target, which is the spike protein on the SARS-CoV-2 coronavirus. So, the coronavirus gets its name because all these spikes stick out and they look sort of like a solar corona. Well, the spike is actually there to bind to its receptor on a target cell. So it has a function for the virus, but that's what the immune system attacks. And so we want it to understand, how do antibodies recognize it? So Christopher, in the lab was totally shut down at that point, except for COVID-19 research. So Christopher was able, with the help of a lot of people in my lab and sometimes even other labs, to just immediately solve all these cryo-electron microscopy structures, which normally we couldn't have done so fast, but most of the people, we have a cryo-electron microscopy facility that's shared across Caltech's campus. But we don't get exclusive use of it, right? But during the pandemic, we were one of the only labs that had a COVID-19 project. So Christopher could really use this. And so he did a lot of these structures. He classified them into different ways that antibodies recognize and then neutralize. So then we thought, well, Christopher classified recognition on the receptor binding domain of the spike. And I'm going to probably slip up and call that RBD. So it's just the part that binds to the host receptor. Okay, so most of the antibodies are directed against that because they just block the receptor binding. So we were thinking that, well, that's great, but these are going to be effective antibodies against SARS-CoV-2, and Michelle Nussenswag has put 2 of them into people now. So clinical trials with 2 of these. They aren't being used except for the trials, but eventually they'll be out there for people to have an option to take if they are infected. But we were thinking that we really want to get antibodies that bind to a more conserved region of this receptor binding domain, not the part that blocks receptor binding. So we plotted how conserved is this part across lots of different coronaviruses? Because I think everybody should have woken up to the fact that this is the 3rd outbreak or pandemic we've had since 2000. We've had SARS, and then we've had MERS, and now we have SARS-CoV-2. And unless we do something, we're going to have SARS-CoV-3 and SARS-CoV-4. And these are likely to come in through bats the way SARS-CoV-2 and SARS and MERS did. And so we just have to have, what I'd like to have is a pan coronavirus vaccine so that it would protect you against SARS-CoV-2 now, but also potentially against other strains. So what Alex Cohen did, who's a graduate student, was he just made these particles, which we can make in the lab. They're small, so they're called nanoparticles, and we attached the receptor binding domains, or RBDs, from lots of different coronaviruses. So people have gone out there and sequenced coronaviruses from bat caves around the world. And I think we have a pangolin RBD on the nanoparticle as well. Like, the sequences that were available, we read the literature, figured out which ones might be a pandemic threat, and attached them to the nanoparticle. And we've done immunizations in mice, and it appears to induce a cross-reactive protective response. And so I'm hoping that could be a vaccine now for SARS-CoV-2, and it might protect against future viruses that might infect us by emerging from their animal hosts.
That's absolutely amazing. I mean, it's fascinating. And so these beta coronaviruses that are on the mosaic nanoparticles, the fact that the mice have they've created antibodies that weren't even represented in the nanoparticle. How can this happen? Is it because of the similar shape, the similar tertiary structure?
Yeah, so what we were most happy about was that we put RBDs from 8 different beta coronaviruses on what we called Mosaic 8. But we deliberately did not put an RBD from conventional SARS because we wanted to ask, can we raise a neutralizing response against SARS? And the answer was yes. So that would imply that if something out there that we don't know about comes in, maybe we could neutralize it. But of course, we can't do those experiments because we don't know what might come in. Yeah. So we're using conventional SARS as the proxy for that. So how can that happen? It's cross-reactivity. And that's what we are hoping to achieve, but we don't know the mechanism of that. To get the mechanism, we need to solve a lot of structures of monoclonal antibodies derived from these immunized animals. So we're trying to do that now.
That's amazing. And that is all my questions answered. You've answered different questions in other answers. And so it's amazing. So thank you so much for joining me today. And I hope to see you at some point, maybe in the future. And yes, just thank you so much. And I'll end the recording and we can have chat as well at the end. So thank you everyone for listening.